Okay, good afternoon, everybody. This is session 331, the Emerging Concepts series. Is it all in the vessels? Understanding the role of vascular system in dementia, including the impact of COVID. I'm Sudha Seshadri, and my co-chair is Professor Kasi Adakola. We will have questions at the end of all four presentations. We will be taking questions both from online, either through the app or through the website, as well as in the hall. Want to assure everybody that there is a whole 45-minute session tomorrow morning where we can continue with the questions and answers. So let's start. Introduce our first speaker?
Yes, so the first speaker is going to be a virtual speaker, and it's a great pleasure for me to introduce our friend and colleague, Dr. Zlokovic, who comes to us from the University of Southern California in the United States.
Good morning. I wish that I'm with you this time in person in the beautiful city of Amsterdam, but this has to wait for another time. I'll review this morning with you evidence from animal and human studies showing that subtle BBB breakdowns, leaks, and dysfunction can lead over time to neuronal and synaptic dysfunction and initiate neurodegenerative process and cognitive impairment. I will share our published and unpublished data showing that gene therapy, cell therapy, biologics, and small molecules directed at the BBB and stabilizing the BBB function can slow down and alter the course of neurodegenerative disorder. I am scientific founder of ZZ Biotech, company with mission to develop new therapeutics for stroke and neurological disorders. But there will be no presentation of any data from the company or material produced by the company. Blood-brain barrier prevents entry into the brain of blood-derived toxic products, microorganisms, and cells. It's represented by endothelial monolayer, and the interaction with pericyte mural cells on the capillaries maintain the blood-brain barrier function and integrity. Blood-brain barrier dysfunction and breakdown and small vessel disease is found in more than 50% of all dementias, including Alzheimer's disease. The key questions that I've been working on for a number of years is what is the role of blood-brain barrier dysfunction in neurodegeneration, cognitive impairment, and AD? A number of animal studies finding in rare human monogenic neurological disorders and findings showing that BBB breakdown is an early biomarker of human cognitive dysfunction led to the theory that blood-brain barrier leaks lead to neuronal and synaptic dysfunction, injury, and can initiate neurodegenerative process. It can happen by many pathways: red blood cell extravasation and toxic iron species, induce reactive oxygen species, and stress. Fibrinogen activates microglia inflammatory response and leads to retraction of neurites. Thrombin is directly neurotoxic. Plasmin degrades extracellular matrix. Albumin leads to edema and activates TGF-beta pathway in astrocytes that is neurotoxic. The anti-antibodies can form against neuronal receptors and transporter where BBB is open, and also a loss of neurotrophic support when pericytes are lost. The blood-brain barrier is an ecosystem of the brain, like what the ozone layer is for the Earth. It's a metabolic organ of the brain, maintains chemical composition necessary for normal neuronal functioning, and I'm going to give you some examples of what loss of blood-brain barrier transporters and receptors can do. Let's start with the glutamate transporter. We transport energy metabolism sugar into the brain and glucose. Lack of glucose transporter, partial lack, haploinsufficiency in a mouse model leads to blood-brain barrier breakdown in 2 weeks. And then during 6 months, there is neuronal dysfunction, as shown here, by what cortical membrane potential responses to high limb stimulation that are completely distorted. And then this leads to overt loss of neurons in hippocampus and microcephaly, of 4%, similar as in GLUT1 deficiency syndrome in humans. So that's an example when a specific endothelial glucose transporter that is only expressed in endothelium, GLUT1 isoform, leads to these changes in neuronal function. The other example I'm going to give you is lipoprotein receptor LRP1. It's an ApoE receptor, major A beta and alpha synuclein clearance receptor, and a receptor for tau. So mice lacking endothelial LRP1, as have recently shown, have blood-brain barrier breakdown because of activation of CPA MMP9 BBB degrading and tissue analysis. And interestingly, neurons remain fine and resilient over the first 2 months of barrier open, but at 4 months, there is a loss of neurons both in cortex and hippocampus. It's a loss of neurons and also accompanied by neuronal loss in behavior, as shown here by null recognition test. Interestingly, gene therapy directed at the blood-brain barrier with AAV2-BR1 endothelial-specific LRP1 delivery leads to recovery of the blood-brain barrier, partial recovery, especially in places over here in green. The blood-brain barrier is intact, and you can see fibrinogen leaking on the other side when the LRP is expressed. And this is achieved by about 45% transduction of the vessels from periphery from AAV injections. Interestingly, this slows down neurodegenerative process. This injection was done in 3 months and results at 4 months in relatively normal levels of neurons and also normal numbers and also normal behavior. It's an example that gene therapy directed at the BBB can lead to reversal. The best example of causal pathogenic link between BBB dysfunction and neurologic disease are human rare monogenic disorders. There are about 20 of those when genetic defects are found only at the BBB and BBB-associated vascular cells, and neurologic disease develops. So this is GLUT1 deficiency syndrome in endothelial cells, MSFD2A, missense mutation lead microcephaly, AMS2A syndrome, psychomotor retardation, and many others. So it is well known that in Alzheimer's disease, there is blood-brain barrier breakdown, as shown by neuropathological studies, and this is showing So we developed a few years ago a DCMRI technique to study BBB integrity in the living human brain, and we showed progressive BBB breakdown in the hippocampus during normal aging and accelerated breakdown actually in MCI. So the question here was, is that related to amyloid beta or tau that are both vasculotoxic? We are talking about early stage when there is no cognitive impairment or there is very little cognitive impairment or some cognitive impairment CDR point. Using accepted cutoff values for A beta and tau, we were able to demonstrate that really this blood-brain barrier changes as shown here are not dependent on A beta positivity or tau positivity. And since ApoE4 gene is the major susceptibility factor for Alzheimer's disease, in a follow-up studies that we published in Nature was shown that ApoE4 gene accelerates BBB breakdown. As you can see here from the maps, this is much lower color. And this is also further accelerated by cognitive impairment, CDR.5 stage. And as you can see, quantifications here show progressive blood-brain barrier breakdown in ApoE4 carrier, not dependent, again, of A beta and tau, by CSF and also by PET. More recent, a published study for the lab, we were asking what is the interaction between blood flow and blood-brain barrier breakdown. And so we studied in memory regions, executive regions, and others, and we showed there is in posterior cingulate gyrus, there is reduction in the blood flow in as we shown. So the question was, do white matter tracts originated from these memory regions that have BBB breakdown or CBF reductions, do they undergo structural changes? So we studied uncinate fasciculus, actually that was by actually Ioannis Papas, and in cingulum and together with the nation, they showed that low FA in UF and cingulum that indicates disrupted tracts happens in ApoE4 carriers, as shown here, both for UF and for cingulum. And this is, this red region shows statistically significant changes in these participants. The next question was, do BBB and CBF changes in these memory regions predict structural changes in Y-matter tracts originating from these regions? And as shown over here, the UF, FA, and in cingulum changes, whether they predict cognitive impairment CDR 0.5 versus 0. And as shown in these cross-sectional studies, they do predict it in ApoE4 carriers, but not in ApoE3 carriers. Earlier studies from our group have shown activation of cyclophilin A MMP9 pathways that degrades blood-brain barrier. An early paper in Nature 2012 confirmed in humans in Nature 2020 by CSF analysis. And then blockade of this pathway in a later stage in ApoE4, transgenic mouse model, as you can see here, treated with cyclophilin A inhibitor, reduces blood-brain barrier changes, and more importantly, leads also to slowdown of neuronal degeneration, as shown here by Debia treatment. So then again, action on the blood-brain barrier with small molecules can lead to improvement of neurological changes. Now, we studied also pericytes, and the pericyte marker as PDGFR-beta. As you know, pericytes maintain BBB integrity, clearance function, and neurotrophic function. And there are participants with high level of PDGFR-beta in the CSF and with lower level of PDGFR-beta in CSF. And we asked the question, is it related to apogenetic risk? In a paper that was published in Nature a couple of years ago, it's shown that high CSF levels, shown here in blue, predicts cognitive decline and mental status in ApoE4, but non-ApoE carrier. Unpublished data and plasma as PDGFR-beta showed that similar thing happens in ApoE4 carrier with their plasma values, and this increase know that we need to generate iPSC-derived pericytes, and we did that through NCC step, as you can see here. And then we use liquid chromatography mass spectrometry to see their composition. In 96, this is quantitative study, 96%, there were no changes in proteins between stem cell-induced pericytes and PCs, and also in phosphopeptide in phosphorylation. That applies to the major categories of canonical pericyte marker, cell adhesion proteins, tight junction proteins, and also transcription factor and protein kinases, and human genome with the protein. And also human genome phosphorylation goes by about 98% similarity. This iPSC-PC can crawl to the blood grafting of iPSC-PC that depends on PDGFR-beta. This integrity prevent neuron loss in this model. So it's a promising future therapy. Now, a few words about 3K3-APC that we developed and is now in a phase 3 stroke trial and completed phase 2 ALS study. This molecule prevents vascular damage. It's anti-inflammatory and prevents neuronal injury and neurodegeneration directly. And here in an earlier studies, we shown that APC also blocks BACE through the NF-kappa-B pathway and blocks amyloid beta production. Now, in unpublished data, here we show that the treatment of E4F mice with APC leads to closing of the blood-brain barrier and increased number of pericytes. And on more mouse, and also improves the proteome in these mice, as shown here in microvessels. The levels of proteins were adjusted to improved by 46%, and their function phosphorylation by 73%. That includes all categories of proteins such as tight junction, adaptor, cell adhesion, cytoskeleton, GTP signaling, and RNA binding. Now I want to spend last couple of minutes on PCOM gene and significant factor for Alzheimer's disease, late onset ID after ApoE, regulates endocytosis, internalization of cell receptors, and etc. It's highly expressed in brain endothelium, and we showed that leads to endocytosis of LRP1 A beta complexes, and these cluttering-coated vesicles then are driven to RAB5 early endosomes, and then to sorting endosome, RAB11, and for exocytosis. PCOM expression is diminished in brain endothelium AD, and PCOM loss from brain endothelium diminishes A beta clearance and worsens A beta pathology, which is reversed by PCOM gene therapy. In drug screening, we identify anti-malaria drug entezonate that upregulates PCOM in brain endothelium, as is shown here. Treatment of FAD PCOM deficient mice with artesunate increases PCOM levels, reduces A beta pathology, and also increases A beta brain to blood clearance and leads to functional improvement. All these effects, protective effects, are lost after PCOM genetic deletion from endothelium. So the key message from this talk is that BBB dysfunction leads to CNS dysfunction, like, for example, human monogenic disorders and animal models. BBB breakdown is an early biomarker of human cognitive dysfunction. ApoE4 carrier develop preclinical BBB breakdown in the hippocampus and mediotemporal lobe, which becomes more severe with cognitive impairment, but is not related to A beta tau pathology. BBB dysfunction can predict cognitive decline in ApoE4 carrier, and therapeutic approaches such as gene therapy, the BBB cell therapy of the BBB small molecules, and biologics such as TRKT-APC can slow down and possibly prevent development of neurodegeneration. I especially thank my wonderful research group that is shown here, and financial support for my studies from the NIA, NINDS, Alzheimer's Association, Cure Alzheimer's Fund, the Duke Foundation, and Open Philanthropy. Thank you very much for your attention.
Thank you. Reminder again to put the questions online. It's now my pleasure to introduce Professor Costantino Iadacola from the Weill Cornell Medical Center at New York.
Thank you very much. It's really a privilege to talk to you today about the role of vascular factors in cognitive impairment and COVID-19. So I'm going to talk to you a little bit about the cerebral blood vessels, the way they are structured and what they do. Then I'm going to talk about the impact of dysfunction of these vessels on diseases associated with cognitive impairment. Then we're going to talk about the role of vascular factors in COVID-19. And if you want to sleep through my presentations, it will be a short one. Essentially, neurovascular health is really critical for the maintenance of brain health, and we need to consider it in therapeutic and preventive approaches to diseases associated with cognitive impairment. So what is the neurovasculome is essentially the entirety of the cerebral vasculatures, from the aortic arch all the way to the smallest venules and capillaries in the brain, and also includes the meninges, which have now, the brain coverings, the meninges, which have now emerged as a critical, important factor in a variety of brain functions. So the circulation of the brain is not only involved in flow regulation, which is very, very important, which includes the delivery of blood flow to the regions of the brain that are active and need the support of more oxygen and glucose. And as we heard, the blood-brain barrier is also another feature of the cerebral blood vessels that regulate the exchange of substrates between the blood and the brain. And the endothelium also produces potent vasodilators agents able to regulate blood flow to the brain independently of anything else. So it's a very, very important cell. However, also another important characteristic is so-called autoregulation, which is the ability of the vessels to maintain more or less constant flow despite fluctuation in blood pressure. So you don't pass out when you stand up and your blood pressure goes down, all right? So the brain tries to keep a balance of blood flow independently of blood pressure. Also, the vessels provide trophic support to neurons and glia, and they also are vital in immune regulation, and we're going to see a little bit about that. But a recently emerged aspect is the involvement of blood vessels in proteostasis. So there are a number of pathways in the brain which are now starting to be developed that suggest that the brain is being cleared of the waste generated by its high metabolic activity through a variety of different mechanisms. Some of these mechanisms rely on the intrinsic ability of the smooth muscle cells to transfer solutes through the interspace between the cells, so-called IPAD mechanism. Another mechanism is straightforward perivascular pathway in which the solutes just travel backward along the vessel wall, and there is also mixing between the interstitial fluid and the CSF. And then there is now the hypothesis that there may be a glymphatic system that washes through the brain and ends to the venous end where it drains into the lymphatics which are present in the coverings of the brain, the meninges. So all these pathways work together to clear the brain of all the junk that is generated through neural activity. This junk is what? A-beta, tau, alpha-synuclein, as far as we know. And they're all kind of clear through these spaces. Now, if there are problems with the circulation of the brain, such as, for example, from genetic factors like ApoE4 that we recently heard, or neurodegenerative pathology, as I will discuss in more detail, or vascular risk factors like high blood pressure, for example, or also changes in lifestyle like, poor diet or low level of exercise or smoking, all of these things will work on the blood vessels to kind of impair its ability to perform all these functions. So what you get is a reduced blood flow, which may lead to hypoxia. You have blood-brain barrier dysfunction, and we've heard how important that is in Alzheimer's disease. And yesterday we heard from Dr. Akasoglu how the fibrinogen getting out of the blood vessels through a faulty blood-brain barrier can have such a pro-inflammatory effect. There will be trophic failure. So the vessels no longer make these vital trophic factors. They maintain the vitality of neurons and glia. There will be altered immune surveillance, and we'll see a little bit about that later, but also there will be altered proteostasis. So stuff gets stuck into the blood vessels, and as a result gets stuck into the brain. And so you have amyloid plaques, you have tau, and so on. So these are all the alterations. So now, is there any evidence that the blood vessels, have something to do with real pathology in Alzheimer's disease? Well, a piece of evidence comes from studies in which there's been a clinical pathological correlation between the clinical diagnosis of the disease of Alzheimer and then what you see at pathology. And a number of studies have been done. Some of you in the audience have participated in this, have generated this data. So if you look at the clinical level in the doctor's office, the majority of patients with dementia is thought to have Alzheimer's disease. However, at the pathology table, only a fraction of them will have classical Alzheimer pathology, that is plaques and tangles. The majority will have a mix between Alzheimer pathology and vascular pathology. So vascular pathology is an important component of the pathological features of Alzheimer's disease and other dementing diseases. Now, another line of evidence comes from a recent proteomic study in which they took the prefrontal cortex from the Ross map cohort in Chicago, and they did the proteomic analysis, and then they correlated the atherosclerosis on the circle of Willis, which is it's a blood vessel at the base of the brain, and the PL vessels which surround the brain, and they correlated that with the dementia, either MCI or Alzheimer. And what they found was that the atherosclerosis of these vessels is an independent pathogenic factor in Alzheimer's disease. And so we're back to the future. We're back to the idea of Alzheimer that first thought, in the 1800s, that one of the major causes of dementia was really hardening of the arteries, which is nothing else than atherosclerosis of those vessels. And what's important for the purpose of this talk is that some of the pathogenic pathways which were inferred from the bioinformatic analysis overlap with those of tau. So you have two factors here. On the opposite end of the spectrum, a neurodegenerative molecule like tau, and the other end you have atherosclerosis, which is a vascular thing, and they both work on neurons through similar pathways, like synaptic dysfunction or RNA processing. So you can see there is a convergence of pathogenic factors on the brain. Now, single-cell RNA sequencing studies have shown that the majority of the Alzheimer risk genes are vascular genes. All right. So this is also another evidence that if you mess with the blood vessels, something bad is going to happen that's going to lead to cognitive impairment. Now, what are the mechanistic basis of this alteration that we see? So let's look at Alzheimer pathology first, particularly amyloid pathology. We first looked at one of the first mice made that overexpresses the amyloid precursor protein, TG2506 mice, at a very early age, like they were young adults. And we looked at the major factors regulating the cerebral circulation, and what we found was that all of these major factors were altered in these mice before they became cognitive impairment, developed cognitive impairment, before they develop plaques or neuroinflammation in the brain, suggesting that vascular dysfunction is an early manifestation of A-beta presence in the brain. And now this has been recently confirmed in patients from the ADNI cohort, in which the vascular biomarkers have emerged as the first biomarkers in the disease progression that occurs in Alzheimer's disease. So in mice, as in Alzheimer, real Alzheimer, there is also this vascular dysfunction which occurs very, very early in the course of the disease. And now, what are the mechanisms of this vascular dysfunction? Where oxidative stress is a major component, which is mediated by A-beta binding in innate immunity receptors. And that's where we get into the neuroimmunology end of things, activating production of free radicals through an enzyme called an ATPase oxidase or NOX2, and as well as an FKB to produce inflammatory changes. So the oxidative stress in turn then leads to activation of a calcium channel, TRPM2 channel, that leads to calcium influx into the endothelium, leading to vascular dysfunction. So for many, many years, we did not know where the radicals were coming from. And the vascular biological literature suggested that perhaps they came from the endothelium or the smooth muscle cells. Well, we found that that's not the case. So these radicals come from myeloid cells closely associated with the cerebral blood vessels called border-associated macrophages that are a population of myeloid cells. They come from the yolk sac like the microglia does, but instead of diffusing into the parenchyma, they get stuck to the blood vessels. They are powerful producers of free radicals because they are enriched with free radical-producing enzyme, and they produce inflammation, and they are located in the perivascular space, which is a critical pathogenic alteration now observed, and as my colleagues will tell you later, very, very much important in all diseases associated with cognitive impairment. So A-beta that is found in the perivascular space as it gets out of the brain acts on the macrophages to activate free radical production, leading to cognitive dysfunction. Now, what about tau, the other major actor in this play of Alzheimer pathology? Well, through my colleagues at Cornell, we found that there are profound alteration also in tau overexpressing mice. We have two kinds of mice here. However, these alterations are limited to functional hyperemia. That is the ability of neurons to increase blood flow. But endothelial cells are working perfectly fine. So this is different from the A-beta mechanism where everything was gone, all right? Endothelial cell reactivity, neurovascular coupling, autoregulation. Here we have a selective dysfunction of the ability of neurons to increase blood flow and getting more blood. And this is due to a reduction, a suppression of production of nitric oxide from interneurons, making the NNOS, neuronal nitric oxide interneurons. And what we found through extensive biochemical studies was that what happens is tau displaces from the soma of the neurons to the postsynaptic density, and then it binds to PSD95, displacing the association with NNOS. So NNOS is no longer able to engage producing nitric oxide in the presence of an NMDA receptor activation. So as a result, you have a nitric oxide deficit, which leads to suppression of functional hyperemia, but because NO is also an inhibitory neurotransmitter, we also have a hyperactivity of the entire neuronal network. So you have, on the one hand, you have the neurons asking for more blood, all right? And then on the other hand, you have the vessels unable to provide this blood to the neurons, resulting essentially in a state of relative hypoxia because of this mismatch, which may be playing a role in the tau accumulation and cognitive deficits. Now, what are the implications, the therapeutic implication of the fact that vessels are important in this disease? Well, one is we've seen that with a beta immunotherapy. So we have seen that antibodies, aducanumab as well as other antibodies, have been shown to do a great job in clearing the plaques, all right? Probably due to the fact that microglia is kind of chewing up the plaques and getting rid of the A-beta. However, if you look at CAA, it's still all there, and it may be even worse of what was before. So there is a problem with the CAA, and when you clear the plaques, but you have... Now, why that may be happening may be the same mechanism that occurs in the ARIA syndrome. So you have the blood vessels are overwhelmed with the A-beta oligomers or monomers or whatever that are released from the microglia because they're not able to metabolize all this A-beta. And so these oligomers migrate to the blood vessels, inducing a state of vascular dysfunction and inflammation that may result in edema, as well as hemorrhages that are classically associated with. And now, we've seen this with a beta immunotherapy, but also with TREM2 immunotherapy. Yesterday, there was a presentation showing that ARIA is also present in TREM2. So you cannot get away from the blood vessels. No matter how you try to clear the A-beta, you're still going to have that problem. Now, another implication for prevention also are present. So studies from Dr. Seshadri as well as others have shown there has been a profound decline in the incidence of dementia over the past several decades. And for a while it's not been known whether it was due to the fact that Alzheimer's pathology may be declining over the decades. And this was recently shown to be not the case. So Alzheimer's pathology is as bad as it is now, as it was in the 60s, all right? But what's different is really vascular pathology, which seems to be profoundly reduced over the past several decades, perhaps except for chronic microinfarcts. So really, this reduction on the pathology probably related to better control of vascular risk factors, hypertension and smoking and so on, is resulting into reduction in perhaps, in dementia incidence. Now, what about COVID-19? As you know, there are many complications involving the brain in COVID-19. Some of them are acute, like, the hypoxia and the coma and seizures. Some of them are the strokes, for which there is an increased incidence about 7-fold. But there are also post-infectious complications, better represented like long COVID or the COVID, the fog, brain fog of COVID. Now, studies done in very selected patients who immediately after they died of the disease, these were studies done in Belgium by the group of Dr. Mombaert, they found that there is no way that the virus gets into the brain. So there are barriers to the virus entering the brain. And so the question is, how does COVID causes brain problems, all right? And so what's resulting from this analysis is that the infection in the blood may affect endothelial cells, affecting producing endothelial inflammation, which then is transferred to cytokines, to the smooth muscle cells, and to pericytes and perivascular macrophages, leading to a cytokine sickness syndrome that then perpetuates in long term a phenomenon of non-resolving inflammation, which is an inflammation which never stops, cannot be kind of terminated, resulting presumably in this condition. In a recent study, which has just been uploaded on the bioarchives, is shown that in patients who died of COVID, in which a punch of the skull was taken, including the meninges, a little bit of brain, they found the spike protein of the virus, which is the most immunogenic part of the virus, present in the meninges, in the vessels, in the bone marrow of the skull, as well as in some brain regions. So the virus is shedding the spike protein, which then finds its way into the brain and may be perpetuating a state of long immune inflammation, low-grade inflammation, that may be the cause of these problems with cognition. So I told you that the neurovasculome is essential for brain health. And then vascular dysfunction is profound. profoundly involved in many diseases associated with cognitive health, including COVID-19. And so if we want to really keep the brain healthy, and if we want to prevent or treat dementia, we got to take care of the blood vessels as well. Thank you very much.
So I now invite...
And the thanks goes to my collaborators and the funding agents who have provided the funding. Thank you very much.
Thank you for that brilliant overview. We now have Professor Raj Kaleria from Newcastle in the United Kingdom.
Thank you. Hopefully you can all hear me. I was told by my mother I was always loud. I never needed a microphone. So thank you, Sudha, and for the organizers to have this wonderful session. I'm sure we'll all benefit. So that's who I am, nothing to declare, and you're free to take pictures, whatever you like. So this is the plan of my talk. Basically, I want to talk from a different angle to what we've just heard, in terms of looking at stroke and vessels. And really to answer that question, is it all in the vessels? And I would say yes, and I'd just sit down. But I'll tell you the whole story, it's okay. And so I want to talk about the COGFAST, the Cognitive Function After Stroke study, and our data from Newcastle and then from Ibadan in Nigeria. Particularly focused on white matter lesions and small vessel disease, and some data on interventions. And then I think what's important, really, is the change in the medial layers of the arteries. Arteriolosclerosis. Not atherosclerosis necessarily, but arteriolosclerosis, and see what I mean. So I think you all know that stroke is a risk for VCI, mild or severe as in vascular dementia. And similarly, one can have dementia and can have strokes. So our first question was really to look at vascular risk factors. And the idea was to look at hypertension. But as you know, to find a clean cohort with hypertension is very difficult in life, anywhere you look, because they'll have comorbidities. So we thought that the best thing to do is to find individuals who have had the first stroke established by CT, and then we can follow them up over time. So 3 months after their stroke, they were enrolled into the study from 6 hospitals in the northeast, in Newcastle area, in northeast of England, and then we followed them. That's baseline. At baseline, they were cognitively intact. So their MMSE scores were something like 28 to 30. And now we really want to see what is the impact of that stroke down the road. And that's really what the COGFAST study is. And here's the basically the protocol of enrollment. And then in parallel, of course, we had aging controls. We also have cohorts in Newcastle, as you will have known before, on individuals who were diagnosed with Alzheimer's disease, vascular dementia, and mixed. And of course, Newcastle is important, as you know, longitudinal studies, and at the end of the day, we want to look at the brain. So we can really compare what we find in the clinic with what we find when we look into the brain. And that's important. Clinical pathological correlation is very important, as you know, and very few centers around the world actually do that in a longitudinal way. And here are the results. Don't get too excited with the glass, but its glass was half full or half empty, and we're not advocating anything if you don't drink. So on your right, you will see that there was improvement in some. 50% chance of another 5 years of life, and better outcomes if they had no risk factors or few risk factors. The glass is half empty, especially if they had more than 2 risk factors. So we're talking about fast track to dementia or death. So the clinical message is so clear, isn't it? Control, strict control of vascular disease and adherence to it. and encourage our patients to do that. And of course, in this longitudinal study, we also found, as was mentioned before, medial temporal lobe atrophy. It's not just related to Alzheimer's disease, but we also see it in these stroke patients, lower CBF and white matter lesions. And as we looked at the trajectories, two important points to make. One is that they remain relatively stable in cognitive function over time, even though they had the stroke, especially if their vascular factors are controlled well. But there's a steep decline 3 years before they develop frank dementia, and vascular dementia in particular. And this is where the clinical message lies. So strict control, again, and that's not really cosmic science. And so you might be wondering, with what happens at the end of the day, what is post-stroke dementia? Basically, most of it is, as we can classify, vascular dementia. As you see, almost 80% of the brains, which we have now collected, 100 brains, so it's a perfect experiment in a sense, where we have half of them who developed dementia before death and half who didn't. And in that way, we can do genetics, we can do blood chemistry, we can do all sorts of things, including they were imaged over time as well as a baseline. And the rest were, of course, mixed dementia. But I want to make this point that microinfarction and small lesions really differentiated the two groups. And here are the data from the pathological data. First of all, on size, PSND, non-demented, post-stroke survivors, and those who are demented. And they have about the same burden of lesions when you think about the size. What was interesting is where the lesions lie. And white matter really differentiated that, as you see here. And small lesions, most of them were smaller than 5 millimeter. So it's just a small lesion that impacts and we see down the road. It's almost something like a virus that hits the brain. And what happens to these individuals? COVID may be, a good example. I'm not going to necessarily talk about COVID, but I think COS has done a great job to us. So most of those brains lie in that classification 2 some years ago, as we showed. Small lesions, multiple lesions, both embolic as well as small vessel disease type and white matter change. And so some years ago, including these brains and a large number of other brains from the brain bank in Newcastle, we scored these in the sense to learn about the natural history of how vessels change and how these lesions progress. So we came up with this classification system. Everybody wants to count everything, isn't it? And that's it. After Braque, we have to do something about vessels. So there you go. And so almost all of the vascular cases lie in that score 6. And there were a large number, in fact, of Alzheimer's disease cases and with score 6, very high. And arteriolosclerosis was a key feature. And when you look at the natural history, arteriolosclerosis followed by amyloid angiopathy were the most common and earliest changes. So looking after our vessels is very important. And then the perivascular spacing and all the other pathologies that we can think of. And this is not something necessarily what I'm saying, but even with this review that we did with Peter Nelson, it shows very clearly in just normal aging people that arteriolosclerosis is an important factor that is aging related. And one can then extend this whole scenario to, of course, what we can see on scans. We can't see arteriolosclerosis on scans. Maybe we measure some sort of way physiologically, but that is there before all these lesions that we define in Alzheimer's disease, including familial AD, as I will illustrate in a moment too. And of course, with that comes the BBB changes. I think somebody earlier today asked, what is the mechanism? Well, if you have hypertension, you've got damaged the endothelium, and that will then lead to other things, including microbleeds and so on. So we looked at the hippocampus, hippocampal formation, atrophy, particularly separated the post-stroke demented from the post-stroke non-demented individuals over time. And then we also looked at the frontal lobe. And what's important about the frontal lobe is that comparing all other groups that were diagnosed with Alzheimer's disease, mixed dementia, vascular dementia at baseline, and the post-stroke demented individuals who, as I mentioned to you, developed dementia over time, they all had atrophy of pyramidal neurons, both in layers 3 and 5, in the dorsolateral prefrontal cortex. So it is completely irrespective or independent of any kind of other pathology, plaques and tangles, that's there. So there's, I think, a whole new arm where atrophy occurs in the brain, and this is where it's sometimes confusing the picture with what's the rest. And that atrophy is very likely instigated by vascular change. Same thing in the medial prefrontal cortex here, and if you want to learn more, there's a poster tomorrow that Dan Jobson will tell you all about. And so we also looked at neurons in these cases, in the frontal lobe, pyramidal neurons, both in layers 3 and 5. And what we see are signatures, really, of basically abnormal metabolism. So just going back to white matter. So over time, we had followed these individuals who had white matter lesions subsequent to stroke. So there were those people who were stable, who didn't develop dementia, and those who were decliners, who started developing cognitive impairment and vascular dementia. And basically, we came to the conclusion that the frontal white matter volumes was an independent predictor of the survival to dementia. And then looking at further the post-mortem material from these individuals to really learn what's happening in the white matter, especially of the decliners. Well, we found profoundly a large number of plasmatodendrocytes, basically dying astrocytes. And these astrocytes, as you know, have end feet to all the vessels, particularly capillaries. And basically they retract the capillaries, the water channel, aquaporin 4 disappears, and they die of autophagy. That's all in this paper and study, and I think illustrated with these. Again, then to ask the question, is this invasive, or is it something that's coming that's non-invasive systemically? And we showed that that's the case, and I'll show the data in a moment. But what was also interesting is that along these capillaries in the white matter, there were activated microglia, as illustrated in the same material. And again, comparing all the other groups, and pericytes, the same issue, comparing all the other groups, you find almost the same changes. Pericytes basically are lost. We talk about PSD, vascular dementia, AD, mixed, it's almost the same picture, all across. And there are similar age groups, etc., and all longitudinally followed. So, of course, then the common factor here is vascular change. And this is just basically the summary. And BBB damage, as you see the bottom line, linked to deep white matter, frontal white matter changes. And we can show this in baboons where we've occluded them, and plasmatodendrocytes occurs again with white matter change. And then one can look at the time course after that occlusion. Similarly, in mice, in BCAS mice, this is a bilateral carotid stenosis model showing also very similar plasmatodendrocytes. And we have not even touched the brain, so it's from the vessels. One other thing that I would like to point out is that if we put those mice 3 hours a day in an enriched environment, they they really reduce the number of strokes that they have, the recurrent strokes after the occlusion. So just now going to the other arm in the last 2 minutes maybe, is about, I want to tell you a little story about our COG-FAST study in Nigeria, and there's something to learn there. We found essentially very similar, or rather, I would say, very similar data from the Nigerian cohort. Medial temporal lobe atrophy, they also developed, those who developed dementia, VCI, 40% would be classified as VAD. Very similar, and we also did imaging there as well. And of course, you might wonder, do they really know apples and oranges? Well, or orientation, and we have to then use tests where the individuals have never put pen to paper to use tests such as these with matchsticks and then be able to score the orientation. So cognition can be very well. But here's the interesting story. One, of course, age we know, but years of education was very important in differentiating those who developed dementia after stroke and those who didn't. But eating fish daily prior to stroke was highly protective, as you see there. So a good message, I think, for all of us. But we might not eat fish, but, you know, something you can extract from it. And then, of course, leafy vegetables are also important in terms of protecting hypertension, incident stroke, and VCI, vascular cognitive impairment. And then with all of this put together, we carefully looked just very recently data from the Colombian cohort with familial PS1 mutations. And we find a profound amount of cerebrovascular pathology in these cases as well. You know, routinely we just look for plaques and tangles, but if you look, you see. I think you understand that expression. So with all that together, I think that we should really redraw this in saying that arteriolosclerosis or small vessel disease really is the first change, and then everything follows. So is it all in the vessels? Yes, absolutely. It's all in the vessels. And protect your vessels. And then, you know, your risk factors, hyperperfusion, white matter lesions, and so all this is a healthy message there. And finally, do you know your blood pressure? This is a rhetorical question. You don't have to answer. Don't raise your hand either. But do you know your blood pressure? Well, look at that. So here's a walking, talking man out in the rural area, and you can see the figures there. So not everybody knows, and he may have very high arteriosclerosis. Okay, so that's just the final conclusion. And I think just to say that arteriosclerosis is an important early change in the natural history of cerebrovascular pathology. Thank you very much for your attention.
So our last speaker is our chair, Dr. Sudha Seshadri from the University of Texas, San Antonio, the role of perivascular spaces in dementia, Alzheimer's disease, and COVID-19.
Thank you. Thank you all. So you've heard enough, I think, to convince you that it is all in the vessels. I'm just starting off by acknowledging my grants and conflicts. I consult for Biogen and Eisai. So the average age at which somebody is diagnosed with clinical dementia is now 85, and at that age, mixed pathology is the norm. At that age, not having any vascular injury is exceptional. And we already know that controlling these vascular risk factors seems to reduce the prevalence of dementia. So how do we understand more about the biology of these vascular mechanisms? We'll be talking today, my role, a little bit about the glymphatic system. We, I think, agree that it's not a question of whether there is vascular injury, but rather how much, what type, and what the impact is going to be. As far back as the 19th century, Verco Robin had shown that there is a lining of the pia around the vessels, around the meninges, and there's a space or a potential space between this. But about 10 years ago, there was a lot of interest when it was shown that in mice during sleep, proteins were cleared quickly along the space. And this was exciting because it answered that question, why do we waste so much of the day sleeping? And it also helped answer the question, what is these perivascular spaces? They're not just anatomical artifacts, they have physiological function. And so you heard already about the glymphatic pathway that not only clears proteins from the brain, abnormal proteins like amyloid, but also brings important nutrients and paravascular molecules, paracrine molecules to the brain. So it's both an inflow and an efflux if it's not working properly. And what are the things that determine how it works? This includes things like inflammation, things like anesthesia, things like sleep disturbances. You already heard about how aquaporin is important. The perivascular macrophages are important. Things like the respiratory and arterial pulsations, which vary depending on whether you're in slow wave sleep or REM sleep, all seem to have an impact. And so the goal is to develop, last year when there was a NINDS summit, one of the goals was, let's find animal models that are relevant to human questions, and let's see if there are human markers based on these animal models so that we can do this in parallel. And for the glymphatic system, you find that if you have mice that are obese or hypertensive, rats that are obese or hypertensive, they have larger ventricles and larger equivalent of perivascular spaces. You see those little white things in the obese mice. And so these are also things that we can image in humans. The STRIVE criteria, the early ones in 2013, as well as the recent criteria, have shown that these are fluid-filled spaces which follow the course of a vessel. If you get them perpendicular, they look like little round or ovoid things. If you get them parallel to the vessel, you see the little arteriole and you see the space, typically not more than 2 millimeters. Interestingly, these seem to precede and are spatially occur at the same spot where later on you see lacunes or microbleeds or the emergence of white matter hyperintensity. And they seem much more frequent in people who have a diagnosis of Alzheimer's disease or who have E4. So this seems like something early to study. One of the challenges is there are so many of them. They're so small. And so while one can do semi-quantitative rating, increasingly we're finding that you can use automated mechanisms, quantitative assessment. And using this in a population study, the multi-ethnic study of aging, or MESA, which has a good representation of African American, Hispanic, and Asian persons, in addition to about 40% European ancestry, Mohamed Habis led a group that showed that basal ganglia and thalamic total volume of perivascular spaces is associated with aging, with systolic blood pressure, and with hypertension. So it's an early marker. And here you can see, even after adjusting for vascular factors and sex and ethnicity, age was a very prominent factor. If you look at these, in this paper, it was using an older semi-quantitative method as described in STRIVE, just counting perivascular enlarged spaces within certain parts of the brain. And in Framingham data, Romero and others showed that this is associated with higher levels of NFL, consistent with the neuronal injury, also associated with a range of inflammatory markers, consistent with inflammation either having a role in the perivascular space enlargement or the perivascular space enlargement in the slowing of removal of proteins, leading to increasing inflammation. This is in the blood. Lighter sleep, not only in the mice, as shown by Mike Nedergaard in that science paper, but in humans, as shown by Andrea and Beryl in the group. They had this presentation yesterday afternoon on sleep in Framingham. And here in 542 people, less total sleep, less slow wave sleep, more light sleep, more of sleep when your O2 sats are dropping, was associated with more enlarged perivascular spaces. And as others have shown as well, in Framingham data as well, as your burden of perivascular spaces increased, your risk of developing dementia increased on follow-up, regardless of whether it was in the basal ganglia, the white matter, or either place. You heard yesterday morning, some of you, that 7 Tesla that is emerging is even more sensitive for looking at and detecting enlarged perivascular spaces. On 3 Tesla, it looks like 1. On 7 Tesla, it looks like 2, you can see. In 7 Tesla, you can clearly see that the enlarged spaces are around little arterioles. You can see, the much clearer. And so one place where this is being used is in using perivascular spaces as a marker of COVID-related dysfunction. We know that severe COVID, and perhaps long-term consequences of COVID are more often seen in people with dementia, and particularly with vascular dementia. This is looking at records of 62 million people. And since we know that COVID is associated with injury to blood vessels and to inflammation, is this reflected in the MR, in small vessel disease, and in enlarged perivascular spaces? When we look at pathology here in the Finnish biobank, you find that people with ApoE4 had more severe COVID-associated pathology in the brain. What about during life? One of the things you heard was that COVID may result in a state of persistent inflammation. So if there is continuing inflammation, can there be changes that emerge well after the acute illness? And in fact, there are a few reports suggesting that enlarged perivascular spaces and white matter, you can see here, these are 6 months after recovery, might actually emerge later. And so this is something that now longitudinal studies like RECOVER are following. This is data from NYU where they quantified enlarged perivascular spaces and found that these were higher in number. This is a small study of 9 and 10 controls and cases, but you can see more enlarged perivascular spaces. So this is being looked at with 7 Tesla. This is also a study with 7 Tesla. So can we use this marker of enlarged perivascular spaces to understand more about the biology of the very early vascular injury? Because we said this happens before some of the other markers. And Marie-Gabrielle Duperon and Stephanie DeBette, who are here, did this very elegant work on over 40,000 people from 18 cohorts, found 24 genome-wide loci, and replicated most of them in a Japanese sample. I won't go into what those genes are, but this is in Nature Medicine. But interestingly, these are genes expressed in the fetal endothelium. These are genes that some of them can lead to leukodystrophy-like syndromes. And this is, the vascular spaces were looked at in the basal ganglia, in the hippocampus, and in the white matter. And you found different sets of genes associated with perivascular burden in these spaces. But there were some genes that overlapped with blood pressure, some that overlapped with white matter hyperintensity or obesity, and some that actually were reflected with changes in circulating protein levels in the blood. And this is unpublished data, but suggests that these are exciting targets. Other ways of looking at it, in collaboration with Dipinder Gill and others, you can say what are the genes that are associated with having more perivascular spaces, and then overlap this with what are the genes that are targeted by drugs that we know. So can these drugs then be repositioned? As you already heard about some of them. And here are some examples. If you look at the white matter perivascular spaces, one drug that targets tinnitus, sulodexide, seemed to come up. You see that it's above that line of significance. If you look at perivascular spaces in the basal ganglia or hippocampus, drugs that target the tau protein seem to be associated with the perivascular spaces. Looking at it differently, if you see what are the gene expression changes associated with PVS, the transcriptomic changes, and then which drugs regulate transcriptome expression of these same genes, you find that statins come up. There's an inverse association. Higher expression perivascular spaces, lower expression with the drug. So simvastatin comes up, a vasodilator comes up, a drug used for pulmonary arterial hypertension comes up. We all have a lot of excitement about these monoclonal antibodies, and we wish it weren't associated with side effects. We need to better understand a natural model. Unfortunately, mice don't develop the area, so we need to have better models. And one of the models in the humans seems to be the cerebral amyloid angiopathy. And squirrel monkeys get cerebral amyloid angiopathy, while rhesus monkeys get amyloid in the parenchyma. And squirrel monkeys, as they age, get ARIA. Just like in cerebral amyloid angiopathy, sometimes you can get spontaneous ARIA with autoantibodies to the amyloid. And so this may be a model where we can better understand. And one word of caution is that when we look at the brains of these squirrel monkeys, there is gliosis. So while ARIA might appear benign initially, one also needs to think of the long-term consequences. We also probably need better markers of the glymphatic clearance, because enlarged perivascular spaces, after all, are just still photographs, right? And so in rats, you find that these are all the different regions where the CSF is cleared. And are there human-based techniques? In rats, for instance, you can put in markers in the ventricles and see how they are cleared along the arteries. And there are some attempts to do this dynamic imaging in humans as well. Here are some pictures where you can see it being cleared over 2, 9, or 24 hours. So I'd like to end by just thanking. As you can see, most of this work is just something I've put together, my colleagues at the Framingham Art Study, in the CHARGE Consortium, in the South Texas ADRC, and in the 70 Brain MRI NeuroCovid Consortium. Thank you. So we now have about 12 minutes for questions. Let's see, are there any online questions?
So I'm going to ask a question. So, let's wait for people to get to the microphone. So I have a question for Dr. Kalaria. So what would you have gotten if you would have included, markers of Alzheimer's pathology in your assessment of the predictability of stroke versus leading to dementia?
You mean in the blood?
Or in the brain, like that.
Oh, right. Obviously, I guess in the living brain, PET would be the one thing. And I just want to say that PIB, very similar study that done by Vincent Mock, they also showed that only about 20% retained amyloid after the stroke. So that was, but I think tau is probably yet to come. However, I should say that it is not unlikely, as we indicated many years ago, that there are probably soluble amount of amyloid as well as soluble forms of tau molecules in the brain. And I think, that really needs a very systematic kind of analysis to look at total extraction from frozen material in a large number of cases, I would think.
Yes, Mike 2.
Thank you, Cyrus Raji, Washington University. Thank you to all the speakers for outstanding presentations and important perspectives. My question is for you, Dr. Yadakola. You showed that paper from the ADNI demonstrating how perfusion on ASL demonstrated the earliest change in Alzheimer's disease. So, extrapolating that information, would you expect that technique could potentially predict changes in individuals who develop ARIA before the actual visually apparent findings are evident on scans?
Yeah, so that's a very interesting question. Certainly, indices of vascular dysfunction will provide insights into who's going to develop ARIA. For example, as you know, ApoE4 individuals have a very heightened, with the TREM2 antibody, 71% of the people, the ApoE4 people, got ARIA. So, the health of the vessels is critical. Now, how to better extract the health of the vessels is a question. You cannot do what we do in a mouse, all right? And vascular reactivity is not the same for every vasodilator. So functional hyperemia, as I've shown you, can be dissociated from another functional vasodilator effect. So we have to be much more nuanced in understanding what goes on at the normal level to be able to extract. But certainly risk factors and the ApoE status to date seems to be the strongest predictors. Thank you for your question. Yeah.
Maybe I'll ask one question, Raj, this is for you from online. Are there any differences between the anterior and posterior vasculature of the brain that could account for neuropathology being more in the frontal lobe with vascular?
It depends where you look. Because the frontal lobe, if you look at the neocortex in the frontal lobe, all over, and similarly in the neocortex of the occipital lobe, there are no differences that we can see quantitatively in terms of capillary density or the microvasculature. But if we look in the white matter, it becomes very interesting. Of course, the white matter, as we all know, has lower density, but those capillaries are 25 to 30% wider, and that we've shown some years ago. So those are the only differences that we see. I think it's a good, and I can see where the question is coming from. I also think that it's the volume of the frontal lobe is much larger than the occipital. And yet, one other difference is that, of course, we see a lot more amyloid angiopathy in the occipital lobe than in the frontal lobe. And that's been shown even along the older cortical layers. We've got actually some unpublished data very recently showing very clearly that that's the case.
Thank you. Mike 6.
Thank you. Thank you very much. I'm Alfred Jamshi from Brain Research Africa Initiative. Thank you very much, Raj and Sudha, for the interesting talks. My question is, at this level of evidence, what do both of you suggest as translation into the clinic and the community? What should I tell my Minister of Public Health from the evidence that we have now in 1 minute? Thank you.
Vascular risk factor control is very important and should be continued throughout life, particularly when cognitive impairment is there. That's not the time to stop. That's the time to keep doing it.
Yeah. And doing it in the preclinical, before, hypertension leads to brain damage, 20 years later, right? So we have to be also proactive. And I would say that, there is a lot of interest from the drug companies now to treat ARIA, to treat patients at high risk because they cannot give the antibodies, all right, with the ApoE4. So to treat them with something. And there was a recent analysis at the ADPD in which they looked at the people who were on statins and whether they develop ARIA or not, but it didn't seem to make much difference. So we still have to find the perfect drug.
And if I may just add to that, is that I think adherence, once a lot of literature now suggesting that they don't adhere to the medication as they should do, and this then brings another issue.
Absolutely. The importance of equity to access as well as convince. Mike 2.
Hi, thank you all for the great talks. I'm Silvia Fossati from Temple University. My question is for Dr. Kalaria, but really to all of you. So based on your findings, do you still think that stroke is a risk factor for Alzheimer's disease, or you think small vessel disease and hypoperfusion are a risk factor for both, and stroke is kind of like an intermediate factor?
Well, we find strokes, yes. So mostly small strokes and micro infarcts that reported in the literature of pathology. Then, of course, the strokes, if recognized with any kind of small lesions, now not looking at microinfarcts per se, are basically removed from the studies, as you know from especially in the living patients. So I think that we're missing something there. But on the other hand, we've published data, 90% of those that come to postmortem or autopsy have some sort of vascular lesion in terms of even a small infarct, and it's there, or microbleeds. So hemosiderin, collection around the capillaries, it's very often finding. We can't do as good a job as the imagers to screen the whole brain, but maybe, it's getting a little better. And I think in the sense that if we sample properly, in a very systematic way, in Newcastle we use large sections, as you might have seen the map from Newcastle. We use really large sections because we reason that what's the point of throwing away the rest of the brain, just having small leads. So we do a whole coronal section, for example, of the frontal lobe, and when you've got it there, you can see a lot more. And we've also looked from 26 regions and narrowed it down to the minimum that tells us about the burden of vascular pathology. And that's in the paper that I showed in Neurology in 2012.
Thank you. I know we are close to 5:30. I would say it's both small vessel disease and the stroke. Preventing the stroke is important. There are two questions online. I'm going to see if Dr. Zlokovic is on, and if not, we'll carry it over and respond by email to the question. So is Dr. Zlokovic on? Do we know? I can't see on this. The question was, and then maybe to both of you as well, regarding the blood-brain barrier changes seen in MCI, do these patients develop Alzheimer later in life as A-beta negative MCI patients? Or do they develop an Alzheimer type picture without developing A-beta or going on to developing A-beta? So Raj, I don't know if you want to take a shot at that. It's for Betzer, but I'm not sure if he's on.
It's a very good question. It's really a searching question. And so one paper comes to mind from, I think neurology was a study from Seoul, from South Korea, and where they looked at vascular risk factors in MCI cohort, diagnosed with MCI, and then they followed them. And so how many of these individuals, if they didn't control vascular risk factors, developed dementia? And it turned out 50-50 who had now frank dementia.
Last question.
Half of them could be classified having AD, and the rest were vascular. So I don't know whether that really answers the question, but I think that's what we need in a longitudinal study.
Yeah. So the paper of Dr. Zlokovic shows that the blood-brain barrier was an independent predictor of the MCI. So even in the one where an amyloid negative, you would anticipate based on that data that they would nevertheless develop. And now, what causes the blood-brain barrier? That remains a question, right? If it's not a beta, I don't know what it is, but we'll leave it up to him.
So we will have more time for questions tomorrow. So one last question.
You were looking at the post-stroke survivors that developed dementia. Did you look at any MCI, that window where they developed dementia from normal cognition? And if you did, were there any biomarkers that you found, anything that you looked at in that sense for the MCI?
Yes, we did. And I didn't show you the data, time limited, but so we had individuals. So in the VIX classification that we published with the Bristol We divided for management purposes and practical purposes, the VCI spectrum into mild and severe. And that severe was equated with what one would define as vascular dementia. So this other end. And they just roughly figures, I would say 50% developed frank dementia over time.